Humans were manipulating the genetics of organisms before we even know what it was. This is how we domesticated corn and livestock. Today, we're going to be taking a look at genetics. So, the science of heredity and variation of traits in living organisms and the study of genes is known as genetics. And it was discovered that the DNA stored in the cells of all organisms is what makes up the code for the inherited traits of all those organisms. So, within the cell, the DNA is stored in the nucleus and it's separated into different pieces called chromosomes. Now, a common analogy is to think of your DNA as an encyclopedia. So there's way too much information to put into one book, so it's broken up onto different chromosomes. So a chromosome is a strand of DNA that contains the instructions for making proteins. And different chromosomes contain different information. The chromosomes become X shaped before the cells divide in the process of mitosis or meiosis. And humans have 23 pairs of chromosomes, or 46 total. Every one of our cells has a complete set of chromosomes, even though it doesn't use all the information. So, the cells that grow our fingernails have information on how to become a liver, but they obviously don't become a liver. So, just like with a cookbook, you'd end up not using all the recipes in the cookbook, but just the ones you need. So, in our eyes, our cells read the DNA that contains two eyes, and in our intestines, our cells read the DNA that relates to intestines. So, genetics is going to be the scientific study of how genes work to determine the characteristics of individuals and to resolve how genetic information gets passed from parent to offspring. When we look at a human karyotype, this is an image of all the chromosomes in one nucleus that have been matched up into their pairs. So there will be two chromosome 1s, two chromosome 2s, and so on into chromosome 23. But chromosome 23 is known as a sex chromosome because they're going to determine the organism's sex. So if you're chromosome XX, you would be a female, and if you're chromosome XY, you're typically going to be a male. So this is what a normal karyotype would look like. You can see there's two chromosomes of every type. They're similar in size and shape and banding pattern. This individual, and if we look at chromosome 23, is going to be a male because they have one chromosome X and one chromosome Y. So if chromosomes are the cookbooks and the gene is the individual recipe for the protein, proteins are responsible for the cooking. So proteins create relatively everything in our body, from our enzymes to the structure of our body. They transport things around our body. They're responsible for creating hormones, for muscle contractions, for our immune and defense systems, as well as for creating energy. And just to remind you that enzymes are chemical components or components that speed up the chemical reactions in our body. So we have different, again, here's the role of the protein. So they support the framework and structure in our body. They move materials within our cells or bodies. They are hormones, which act as signals to coordinate and regulate activities within the body. They allow our muscles to contract and relax and are important in our body's defenses. Now, prior to a child being born, there's a process that you can do called amniocentesis that will look at the karyotype of the unborn child. So what doctors will do is insert a long needle into the amniotic sac to draw out some fluid, and this contains cells from the developing baby. So these cells are then examined for genetic abnormalities such as additional chromosomes like Down syndrome or any genetic conditions the parents might know that they are carriers for. So mitosis, meiosis, and selective breeding are three things that we know about genetics. So we know about mitosis and meiosis, and selective breeding is what we do with that information. So mitosis occurs in autosomal cells or our regular body cells, like our skin and our organs. The daughter cells created through mitosis are identical to the original parent cell. They have the exact same number of chromosomes and the exact same chromosomes in each daughter cell. All autosomal cells have 46 chromosomes before division and 46 chromosomes after division. And then because each original cell and daughter cell have two copies of each chromosome, they're referred to as diploid cells. So we're 2N, we have two copies of each chromosome, 1 through 23. And since the pairs of chromosomes have genes for the same characteristics at the same location, they're going to be referred to as homologous chromosomes. So when we looked at that karyotype, we have two chromosome 1s, they're homologous chromosomes. So humans grow, build, and repair through mitosis, but some organisms are able to produce another organism through mitosis, and this is known as asexual reproduction. And this is things like budding in hydra or creating runners in strawberries. So, and the parents are then creating genetically identical offspring to themselves. So, meiosis is the process by which organisms reproduce through sexual reproduction. So, before we can reproduce through sexual reproduction, we have to create gametes, which are cells that have half of the genetic material of the parent cell. Because then, after fertilization, we want that cell to have the same amount of chromosomes as a somatic cell. So, we create gametes such as sperm or eggs, or pollen and eggs in plants. So the process by which this happens is meiosis. So in meiosis, we start with the replicated DNA, which then pairs into its homologous chromosomes. Those homologous pairs are going to then separate, and then we're even going to break up the pairs of sister chromatids. So each one of the gametes is left with one chromosome, or half the original amount of genetic material from the parent cell. So we call these cells haploid because they have half of the genetic material as the parent cell. When you look in humans, the sperm or egg cells are haploid, or just simply N, because they have half the number of chromosomes compared to autosomal or, say, skin cells. This is because the egg needs to be fertilized by the sperm, and we want those two together to then be a diploid cell with half the genetic material coming from the egg and half the genetic material coming from the sperm. Also, each chromosome in the gamete is able to differ from the original cell due to a process called crossing over. And then this is when those homologous chromosomes are laying on top of each other, kind of like cooked and sticky spaghetti. Some genetic material is able to transfer between the maternal and paternal chromosomes during this process. So this is why we have the wide amount of variation that we do when animals and humans use sexual reproduction. One way to remember the difference between mitosis and meiosis is can be remembered as mitosis. So we produce two diploid cells that are 2N, where meiosis can be remembered as meiosis, where we produce haploid cells with one copy of each chromosome. So in meiosis, they have one chromosome. Mitosis, they have two chromosomes, two copies of each chromosome. After meiosis occurs, we need to come up with a process by which those two gametes are joined, and this would be fertilization. So during fertilization, the sperm and the egg in humans, or the egg and the pollen in plants, are going to join together. And that fused set is now going to contain two sets of chromosomes, the same as in autosomal cells. From the point of fertilization onward, those cells are going to grow and develop through mitosis from that single celled organism to a multi-celled plant or human. Now, there are some advantages of sexual reproduction. One of them is that there's more diverse offspring produced. And because we have more diverse offspring and a more diverse population, we're more likely to survive extreme conditions or environmental changes. There are more than 70 trillion ways for the gametes from a mother and father to join and create a new human being, which is why siblings, although they are similar, are often quite different from one another. So we take our knowledge of mitosis and meiosis, and we do something called selective breeding. Now we do this in dogs and cattle and plants, for example. So this is when we choose individuals with useful characteristics or characteristics that we desire to create a plant or animal stock with those characteristics. So First Nations people use selective breeding to develop corn from a type of wild grass. So corn is a domestic plant created from wild grasses. Now we can look at the selective breeding in cows. And so we want cows that have strong maternal instinct and a high growth rate. So we're going to breed those together. And then the offspring that could have average instinct or high growth rate, average instinct and average growth rate, strong instinct and average growth rate. But we might have one that has strong maternal instinct and high growth rate, where we're going to select that cow for future breeding. And the other ones might be sent to be food stock. So Gregor Mendel was a monk who was the first person to undertake studies into the inheritance of characteristics. And he studied pea plants and he found some interesting patterns. One was that plants could produce seeds that are either green or yellow, round or wrinkled. Plants could have purple or yellow flowers. We could have inflated pods or pinched pods, yellow or green pods, axial or terminal flowers, or have longs or short stems. And these traits all occurred in an either or pattern. So one of the things he discovered was that when he bred a true breeding purple flower plant with a true breeding white flower plant, that first generation all had purple flowers. However, after he bred that purple generation of the first offspring, the second generation had about 75% purple flowers and 25% white flowers. And this was a very standard ratio that occurred when he was crossbreeding plants. So crossbreeding plants is when you transfer pollen between two genetically different plants. And self-pollinating plants are when you take pollen from one plant to the pollen from the male part of the plant to the female part of the plant. So this is how we can ensure there's true breeding lines of plants, is if you self-pollinate them all. And so Mendel would use cross-pollination to determine the ratio of certain traits that we developed, and he would use self-pollinating to make sure traits stayed in a certain ratio. So it would only, if he self-pollinated a purple plant, he would only get, say, purple plants. So that's our introduction to genetics and inheritance. I hope you enjoyed today's lesson.